Hi friends, welcome to my video. In this video, we are going to see about Griffith Experiment. Transformation in bacteria was first demonstrated in 1928 by an English medical officer, Frederick Griffith. Griffith was researching about the distribution of pneumococcal types. Griffith conducted a series of experiments using Streptococcus pneumoniae and mice. In his experiment, which is popularly known as Griffith Experiment, Griffith used two related strains of bacteria known as R and S strain. Let's see what is R strain. When grown in a petri dish, the R strain formed colonies that had a rough appearance. R stands for rough. The R strains were non-virulent. That means they did not cause disease when injected into a mouse. Let's see what is S strain. When grown in a petri dish, the S strain formed colonies that had a smooth appearance. S stands for smooth. The smooth appearance was due to the presence of a polysaccharide capsule. This capsule protected the S strain from the mouse immune system, making them virulent. That means when injected mice with this S strain, the mice developed pneumonia and died. Friends, up to this we learned what is R strain and what is S strain. Next, we are going to say the outline of Griffith experiment. Griffith took two groups of mice. For the first group of the mice, he injected R strain, and for the second group of the mice, he injected S strain. Mice injected with R strain lived, and mice injected with S strain died. We know the reason, because R is non-virulent and S is virulent. In the next part of the experiment, the mice were injected with EAT-killed S strain. The mice all lived. Here also we know the reason, because he used EAT-killed virulent strain. In the third part of the experiment, the mice were injected with a mixture of EAT-killed S strain and live R strain. From the previous experiment, we learned that the strains used alone were unable to cause disease. But surprisingly, the mice all died. Blood sampling showed that the blood of the dead mice contained both live S strain and R strain. The original virulent strain was EAT inactivated. Griffith concluded that the non-virulent R strain had transformed into the virulent S strain. Griffith postulated that information, which he called transforming principle, could somehow be transferred between different strains of bacteria and converted the R strain into S strain. We should remember one important point here, that this was long before the discovery of DNA as a genetic material. Griffith was not able to identify the chemical nature of this transforming principle. The nature of the transforming principle was identified as DNA by Avery, McLeod and McCarthy in 1944. Thus, Griffith experiment led to the discovery of DNA as the genetic material. While explaining the Griffith experiment, I told you Griffith used EAT-killed S strain and live R strain. So you may ask me how DNA from EAT-killed S strain convert R strain into S strain. The answer is that DNA molecules can renature after EAT treatment. Renaturation is the reconstruction of a nucleic acid to their original form, especially after denaturation. I'm concluding this video here and see you soon in my next video. Thank you, friends.